cDNA library, say it is a completely unknown organism. Say people landed in moon, and then they found there is life form, came back with it. Is there any use of cDNA library there? Anyone?
So we can generate cDNA library, but use.
So should I try once?
What is the use?
Excuse me, sir. Sir, can you please repeat your question?
No, Promit raised hand. I would ask Promit. Promit.
Like the example you gave for Mariana Trench, so we wouldn't know if, like for example, yeast genome doesn't have any introns, so we wouldn't know if the organism we found in the trench does it have an intron or not. So in that case, we wouldn't be able to exactly create a library. So it would be better if we create mRNA, because all those non-coding regions between the exons will be removed, and so we'll have only the those regions which is complementary to the coding sequence.
Okay. Let me pose the problem to you. See, like I am trying to make you think, and think like an experimental biologist. So I will draft the problem for you, and every sentence that I put in a question is important. There is I do not do those things, just put some random sentences to confuse you. I do not do that, okay? Every sentence has a meaning. So now say that you got an organism from the depth of Mariana Trench, and some initial experiments were done with it, and you have reasons to believe that this organism behaves very similarly to E. coli. Its genome, its sequence might also look very similar to E. coli. And now you want to know if this organism has an equivalent of hexokinase. How would you do it? Promit, you want to try? Try it.
Yes, sir. I'm just thinking about it.
Okay.
So, do you want me to answer?
Yeah, please.
So the purpose of either genomic DNA library or cDNA library is to identify genes that are there in the organism. So if the organism, for instance, if we feel that the novel organism will have introns in its genome, then we would go ahead with making a cDNA library. And say we will identify, as you said, hexokinase in the organism. So we would first we would first isolate the RNA from the novel organism, convert it to cDNA, then clone all the cDNA fragment in the plants, and then we transform the ligation in E. coli, and then we go for plating. After we plate the E. coli, we would observe some colonies growing in the E. coli plate. So, so each of those colonies will harbor a plasmid containing the cDNA, different cDNA fragments. Those fragments are basically the genes. So, so it is possible one of the colonies may contain the gene of interest. So what we need to do is that we need to, since the hexokinase gene is very conserved, we can prepare probes for the hexokinase gene. So degenerate
Kind of
probes we can
probes?
prepare.
What kind of probes?
So degenerate probes. So I mean, we know the protein sequence for the hexokinase, so we can reverse translate it so that we can get the nucleotide sequence of a short fragment of hexokinase gene that is preserved. But we know that because the codons are degenerate, so there are 2 or 3 codons that can code for one amino acid, right? So we need to see, and we make probes accordingly, and then we see which of the probes is hybridizing to the nitrocellulose.
How would you
sorry, sir.
what will be the readout?
Hello.
What will be the readout for checking hybridization? What will be the readout for checking hybridization?
We use probes, right? We use radioactive probes, so the probe will.
Very good. Very good. Take it. I got it. So the fundamental issue is that cDNA library is your method for having the cDNA counterpart of every mRNA as a collection. Okay, so that is the first thing. Now that helps you in getting to know the sequence of different mRNAs. Okay, if you know the cDNA sequence, you get to know the mRNA sequence. So that is the first thing.
Sir, another thing is maybe
sometimes, yeah, go ahead.
Sir, another thing is dealing with
Go
the
ahead,
mRNA is hello, sir. Sir, another thing is dealing with mRNA is tedious because it has a secondary structure. So when it forms secondary structure, then we cannot use probe properly. Then we can make it so cDNA, so we can easily access that cDNA rather than the mRNA. That's why.
No, not only that, Kuna. The problem is more fundamental. The problem is more fundamental. There is no way you can separate one mRNA from the other in a solution. Therefore, you cannot sequence the RNAs. Okay. Therefore, you have to convert them to cDNAs, and by definition, then you clone them, so that becomes your cDNA library. Then only you can do sequencing. You cannot sequence a population of RNA, right?
Yes, sir.
That's the other problem. But there is, yes, Shreya, go ahead.
Sir, here we are discussing about hexokinase, which is like a conserved enzyme which is found across organism. But as we have taken the example from Mariana Trench, so there may be some proteins which help that organism thrive under such extreme pressure and temperature, which may be very novel to that organism. So here we do not have a reference to build a probe for such identifying such proteins. So then, how will we
Okay, I will give you the answer to that as well. Okay, you give me 5-7 minutes. Let me bring all my students to the same level. Then I promise I will respond. I will give you a very good answer. Okay. So, Nageshwar, of everything that you said, the only thing that I would like to correct you is that you do not necessarily start with a degenerate probe. Okay. A degenerate probe is a last resort if you do not get any response, because degenerate probe has many nuances. As you can imagine that there can be so many variants of the same sequence giving the same amino acid sequence. So degenerate probe has its own problems. But the advantage with hybridization is that even if there is the similarity over 100 base pair stretch with a couple of dissimilarities, you will still get hybridization to work.
Yes.
Okay, so that's why.
Okay.
Shayan, you were saying something?
No, sir. I was
Okay.
asking something. I don't.
Okay. Now, let me ask the other question, which will take me closer to Shreya's question. That imagine, just for the sake of simplicity, we are sticking to yeast only. Okay, we are not going to higher organism right now. Imagine that we are back to the same problem, gal minus mutants. And I have forbade you to use genomic DNA library. Okay. Now you have to use cDNA library. How will you go about finding the gene responsible for growth in presence of galactose?
Sir, can you repeat the question once again?
Yeah, that you learned how we do complementation with genomic DNA library, right?
Yes.
Now imagine that I have explicitly stated you cannot use genomic DNA library. जैसे मूवी डायरेक्टर में डायरेक्टर अगर चाहते हैं तो 100 गोलियों से भी अमिताभ बच्चन नहीं मरते हैं, कभी कभी एक गोली से भी मर जाते हैं. So examiner is supreme here. So I do not allow you to use genomic DNA library. You have to use cDNA library. How will you do it? You have to do complementation analysis, but this time with cDNA library. How will you do it? Yeah, Ayush, go ahead.
So what we will do, sir, firstly, the question is we are using a simple organism, right? From which we are creating
Yeast.
cDNA library.
Yeast.
Yeast only, okay. Okay. So what we will do is we will isolate total mRNA out of the RNA content that is there in the yeast. For that, what we will do is we will use poly-T columns, columns which are coated with poly-T, and they will preferentially immobilize mRNA out of the total RNA content. For that, we will use a simple kit and we will isolate total mRNA using the kit and like trizol method or something. And after isolating total mRNA, we will convert it into cDNA, and then we will partially digest it. The cDNA, what we will get, we will do partial digestion, and after getting the fragments, the part of the fragments, I'm elaborating more of the steps which are used to create a cDNA library.
Sure. I like that. I like that. Go ahead.
And then we will choose the fragments one by one, clone it into the vectors, and then transform it into the yeast and into an yeast strain, haploid yeast strain, which is gal negative. And then we will check for the growth of yeast on galactose as a carbon source, which of the fragment when transformed into the yeast will result in growth of that particular strain. And that's how we will isolate it and sequence it and know that which gene is involved.
Okay. Okay. I get your answer. Now, let me share my screen. Okay, you can see my screen, right?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
So this is mRNA. This is the cap structure. This is poly-A tail, etc. Then, as Ayush said, we will be 5 prime, 3 prime. We will be using an oligo DT so that it elongates in this direction, and then it is 3 prime. Then we will add there are several methods. In one method, you can add adapter to the RNA itself using RNA ligase. You can add adapter to the after doing this first step, you can do a digestion, and then you can amplify the second step. If you see here in this particular example, can you see my lecture slide here, or you still are seeing the blank slide?
The blank slide, sir.
You're seeing the blank slide. Okay. Let us go through the lecture slide another time. Okay. So you see that what is being done here is that in this particular case, you use an RNA ligase. You do your first, so this green line, this is called the first strand cDNA synthesis, okay? Because it is the antisense of the RNA in the form of DNA that is being synthesized. Then you degrade the RNA, okay, by alkali treatment, and then you use a ligase to add this poly G on the 3 prime side, and then we use a poly C for the next step. And because these are artificial sequences, after that you can add your linker, or you can have the linker in this only, so that you have 2 restriction sites and you clone it. Okay, no problem. Now, and there are other methods as well. You can add the adapter right at the stage of the RNA itself, and then you can proceed. So that is also possible. So you can do either one of these. Now, let us go back to the other slide.
So can you just repeat what you said last? My net connection went off actually.
Okay, so what I'm saying is that either you add an adapter sequence after the first strand synthesis, or you add the adapter at the RNA level itself. Okay, both kinds of enzymes are possible, available to add the adapter. Okay, so what you will end up doing, so this is your 5 prime and this is your 3 prime, and you will add a known sequence as adapter. Then you will design another primer. You will design another primer which will be 5 prime here, but it will anneal with this part, and then it will extend on this part, and then it will finish here, and you will have, and you can with this double-stranded DNA, you can add another linker which will have restriction site here. This will have restriction site here. All of this is possible. Okay. Now, when you say that I will transform the gal strain with the cDNA library, and at least 1 or 2 colonies, I will see that the cDNA, like the transformed cell, is able to grow in the absence of galactose. What is the minimum condition that must be satisfied for the gal minus strain to be able to grow in absence of galactose? Ankit, bolo aap. Listen to my question again.
Yes, sir.
I have transformed gal minus strain with cDNA library, and you expect couple of colonies which are transformed to grow in the absence of galactose. What is the minimum condition that must be satisfied for this to be true?
Sir, the cDNA which has been transformed should be the cDNA which is responsible for the gal metabolizing activity. The galactose metabolizing activity. That is
given, I will say that itself will not do the trick.
okay.
That by itself may not do the trick. You have to do something else.
Sir, we have to select the yeast cell. If the yeast cell is negative, if the yeast cell has a mutation in the URA3 gene, then
that is
yes,
also there. So that's the only way you can grow the transformants, right? So that is not a problem.
Yes. Okay.
For the yeast cell to be able to grow in absence of galactose, what has to happen?
Sir, can I try? Yeah. So I think
Please,
for galactose metabolism, there is like multiple genes involved. Like there is an enzyme for galactose intake, and there is an enzyme for galactose digestion. So I think we should create combination of those cDNA libraries within the same plasmid.
No, absolutely wrong.
So
Because you know that only one gene is mutated, right? So why do you have to offer a combo? If you can offer,
so we have to make sure like the dominant negative mutation here. So what if the mutated gene?
like right now, let's take it as recessive, no dominant negative.
Okay, all right.
First let's learn the baby steps, then we will go for running and high jump.
Sir, I would like to get a clarification. Since you are saying that the strain should grow in the absence of galactose, is it not like it should grow in the presence of galactose and it should be able to metabolize the galactose? Or the question is, it should be able to grow in absence of galactose?
Oh, sorry. I am wrong. It should be, I am absolutely wrong. It should be able to grow in the absence of glucose, but only in the presence of galactose.
Galactose, yes.
Like galactose is the sole carbon source.
Okay.
I misspoke, but I meant it that way. So you are really looking at the galactose, metabolizing genes. So what will be the minimum condition that must be satisfied?
Sir, the insert, the cDNA we have insert to the expression host should be in the frame that it should be expressed.
Yes.
So that is the first thing that you should say, right? That unless the mutant gene is expressed from the cDNA, the plasmid containing the cDNA, there is no possibility of rescue, right?
Yes.
So now let us think about it, that what should that plasmid minimally contain so that it is expressed. I will go
The
from
ribosome binding site.
एग्जाम में -25 से भी चालू हो सकता है.
Timer for that.
Is the expression happening in bacteria or in yeast?
In yeast, sir.
Focus is
It should be
Exactly.
ARS. ARS
So
sequence should be there.
Sir, it should having some
सब
promoters.
बहुत खतरनाक मोड़ पे आप जा रहे हो. ARS has nothing to do with translation. ARS is autonomously replicating sequence. It's a DNA replication sequence signature. It has nothing to do with protein synthesis.
Okay, sir.
So it should have a promoter sequence which is specific for each strain, and then it should also have I mean the gene construct should also contain the 5' and 3' UTR regions, so that ribosomes could sit on those, and then translation machinery could work.
So you have no control over the 5', 3' UTR, right? Because you are making the cDNA from the mRNA using a reverse transcriptase. The 3' UTR will obviously be there because you are starting from here. अरे मैंने तो यहीं पे गड़बड़ कर दिया. हाँ, so because it is starting from here, so 3' UTR will obviously be there. There is no option, right? 5' UTR, you have no control over it, but yes, what you were saying is partly correct, but it is not entirely correct.
Sir, hello, sir.
हाँ, बोलो.
Sir, we have to add the adapter or the linker in such a way that, and having some of the restriction enzyme in that adapter, when you cut with the restriction enzyme and link to the vector expression vector, so that the promoters and the cDNA is in the frame, that it will start when it's transcribed, the mRNA should be in the frame with that promoters. So we have to add and optimize like which type of restriction enzyme we have to add in. Hello, sir.
Okay,
Yes,
I heard you. Puna, what sets the reading frame?
Sir, reading frame is like it. Oh, start codon, stop codon, sir. Open reading frame, sir. It is, sir, open reading frame. Hello, sir.
आप सबका प्रॉब्लम कहाँ पे है पता है?
Test books.
Like in शुद्ध हिंदी, आप सबके फंडा में बहुत सारा अंडा है. That's where the problem is. What sets the reading frame? Can anybody give me an answer? What sets the reading frame?
The first residue that starts with AUG.
Start codon to stop codon.
It starts with start codon and
recognition of AUG sets the reading frame.
Yes, sir.
Wherever the scanning ribosome hits the first AUG, after that, every 3 nucleotides are taken for a new amino acid. Right?
Yes, sir.
Yes, sir.
Yes, sir.
Therefore, therefore, if you are making a cDNA library, cDNA, so in the mRNA, imagine this is where you have the AUG. Okay?
Yes, sir.
So by definition, there cannot be any AUG here. In the 5' UTR, generally speaking, there are exceptions. Let us not worry about the exceptions right now. In the 5' UTR, generally there should not be any AUG. So if your cDNA is like this, which is cloned in a plasmid, someone said promoter, another term is there, we will talk about it later, enhancer. Then the job of the promoter, this I will teach in whole lot detail when I will teach you my transcription. Promoter, do not ascribe it any special power. Promoter does only one thing. It recruits RNA polymerase right there. Okay, and wherever RNA polymerase is recruited, 10 nucleotides or 20 nucleotides, 25 nucleotides from there, transcription starts. Right?
Yes.
And all this part is in the vector. Only this part is what you have cloned, right? The rest is in the vector. The vectors are designed in a manner that there is no AUG in this stretch.
Yes.
Okay?
Yes.
And there is no AUG in the 5' UTR. So from here, the RNA that will be made will have part of the vector sequence, then the RNA sequence, इसको ATG कर देते हैं, and then somewhere over here is the start codon, right? Am I making complete sense?
Yes, sir.
Then the ribosome
Yes, sir.
recruited here at the cap structure, because we are talking about yeast, we are not talking about bacteria, right?
Yes, sir.
In yeast, ribosome is always recruited at the 5' end.
Yes.
It will keep
Yes,
on
sir.
scanning till, like, I should not be drawing the larger subunit here. It will keep on scanning till it hits AUG, right?
Yes, sir. Then it
Yes, sir.
When it hits AUG, that's where the large subunit will bind to it,
binds.
and your reading frame is set.
Yes, sir.
Therefore, in an ideal scenario, you need not worry about the ORF,
Yes, sir.
because AUG is provided from the mRNA itself.
Yes, sir.
Yes, sir.
So,
Okay.
sir, we have to we have the challenge. We have to fix that the transcribed part, then the transcription side, and
Yes.
Yes,
If
sir.
it is eukaryote,
Yes, sir.
all you care for is that the transcription should happen.
Yes, sir.
If it is prokaryote, then you will have to have a mechanism of having a ribosome binding site here.
Yes, sir.
Yes.
Because the mRNA
Yes, sir.
may or may not have a ribosome binding site, so you can have a ribosome binding site there. It is prokaryote. Okay?
Yes, sir. Yes, sir.
Completely
sir.
clear?
Yes.
Okay.
Yes, sir.
Now,
Sir, I have a question regarding this genomic DNA and cDNA library construction. Can I ask it?
Yes.
Yes,
Yes,
sir.
please.
So after we construct this, when we are doing this genomic DNA or cDNA library construction, in one of the steps we transform this ligation mixture into the E. coli cells and we plate them. So let us say we observe some, say some 500 colonies that we have got in the transform plate. Now, if we want to transform this genomic DNA library or cDNA library into yeast cells, do we pick all the 500 different colonies, do 500 plasmid isolations, and then isolate all the 500 plasmids individually, and then
no.
pull together
For
and then
different applications, so for different applications, different way it is done. If it is for complementation assay, the way we have learned so far, you can actually work with the pool.
Okay, so how exactly this pool is constructed and then transformed?
So you can actually grow the colonies and then scrape the colonies into a solution and then grow it together, and then from there you can isolate the plasmid, or you can after transformation, instead of plating them, you can grow them right there. It is little risky, but many people do it.
Okay.
You understand that after transformation you do not plate. You just grow them in presence of ampicillin. That's it.
Okay.
With the assumption that only transformed bacteria will grow.
Yes.
Right?
Okay, sir. I have understood right now. That is because I was wondering, because there were like 500 different genes. So I was wondering if we had to do like
Yeah,
500
it is not
individual
500, it is like
plasmid.
10,000, 30,000 and more.
Okay.
Ayush, you were asking something.
Yes, so it might be a very silly question, but it is
No
important
question
to.
is silly. Every question is important.
Okay, so I wanted to know that in the if we look at the screen in this portion where you have drawn it with the red color, so if we reduce the length of this portion, if we reduce it to like a minimum length, like 2 or 3 bases or whatever minimum we can do, so will the function, will the mRNA form then be functional for translation, or will there be any effect in terms of eukaryotic translation? I want to know this.
Yeah, so ideally it should not hurt because you are making a replica of the actual mRNA. So if the actual mRNA could be translated efficiently, then this one from the vector should also be able to translate efficiently. You do not need any extra sequence coming from the vector. But in reality, what happens is when you are trying to make cDNA library, often reverse transcription can get terminated here, 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 and that's a mess. Okay, like there is no shortcut to those. Sometimes you suffer. Okay, because if you have not been able to reach even up to the AUG, then you can imagine what will happen. Like some other AUG will be chosen as the first codon, and then it might be translating something completely off the frame, or it might use an internal AUG coding for an in-frame methionine, so it will make a truncated protein. All sorts of circus happens there. Okay, and it was quite routine during like when we were trying to do these things.
Okay,
you did not ask another question, that no matter what, before cloning it, you are digesting with some enzyme, right? One enzyme, two enzymes, some enzyme, right?
Yes, sir.
Yes.
There is no guarantee that not a single mRNA or its
AOG
corresponding will not have the restriction site within the what-if, right? So if that is there,
Sir,
then
I did not understand your.
So what you are doing is, if this is your actual cDNA, by design, you are appending a linker here for restriction site 1 and a linker here for restriction site 2, right? That's what you are doing while making the cDNA, right?
Yes, sir.
And then you are digesting with restriction enzyme 1, restriction enzyme 2, and cloning it in the vector that is also digested with restriction enzyme 1 and restriction enzyme 2, right? Is there any guarantee that in this stretch there will be no site for R1 or R2?
No,
there is no such guarantee, right?
But we are methylated, na?
No.
And we are methylating, na, by DNA methylase.
By what?
That we are protecting, na, by extra methyl group.
No, you are not. See, first of all, not all restriction enzymes are susceptible to methylation modification. Okay, there are majority will not care. Second is that, please understand, you are making this by in vitro reverse transcription. Okay, so there is no possibility of methylation happening. If you look at your slide, if you open your slide on the other window, you will see that there is no step for adding methyl to the sequence. In fact, this happens quite often, okay? Certain genes, you will not be able to clone full length for a variety of reasons.
Sir, actually, he's getting confused between two methods. There was one more method in which, in your slide, like the strand was methylated. That is why.
Let me check. I want to be sure that.
So that It is simply the basics of how bacteria protects its own genome from
Yes.
cutting it.
Yes. But that's where, like, upon methylation, it is protected, right?
Yes.
So, which slide are you referring to?
Sir, the topic is cDNA library contains
Oh,
representative.
I see it. I see it. Okay, that is slide number 42.
Yeah, sir.
Right.
Yes, yes, sir.
Yeah.
So in that
Yeah,
there
like
is a step.
So the sixth.
yeah, like this is not really very widely used, and even if it is used, I can tell you from experience. Like, first of all, I have never used it. I have made many cDNA libraries. I have never used it, and even if it is used, I do not know how 100% effective it is. Okay. The more common one nowadays is this one, which is there in slide 43. As you can see that it is basically a commercial source. I have provided the source of the kit also in the bottom of the screen. So that's how it is more often done. Okay, so now before I forget, let me answer Shreya's question. Shreya, because you asked, so I am answering. Now, do not kill me for overkilling, okay? But because you guys are learning, so I am excited. So I will, Shreya, give you an example, which is a little bit more straightforward, okay? Instead of Mariana Trench, we will give you a more straightforward example. Say, you know that there is a protein X present in yeast cerevisiae, which generally gets phosphorylated. Okay. Now, and you know the enzyme. The name of the enzyme is E. Okay. Because it is cerevisiae, let's mark everything as SC. Okay. Now you found a new organism which is very similar to cerevisiae yeast. Okay. You want to find the ESC counterpart in there. Okay. Or, so that is one problem. The second problem, you know that there is a, say in human cells, you know, like we still do not know function of most of the things, right? Say you discovered a protein that is like the protein name, sequence, etc. is known, but you just figured out that that is very important for, say, forming neuronal connections. Okay. And say that you figured out that this happens because there is a protein P, it is mammalian. अच्छा, प्रोटीन का नाम P नहीं हो सकता है. Protein Y, which gets phosphorylated. Okay, I am choosing phosphorylation for a reason. And you know that this phosphorylation is very important for either function. So you want to know which enzyme does the phosphorylation. Okay, how would you do it? So this will be very close to your Mariana Trench question, right?
Yeah.
Because it is not loss of a gene that you are looking at. You are looking at an enzyme that converts a substrate into a product, right?
Yeah.
How would you do it? So the way it is done, it can be done. There are actually papers on similar lines. So imagine that you have constructed a expression library, cDNA expression library, the way we did it in the last slide. Okay? Now you have transformed it. Each plate, say, has some 300 colonies. Okay, and you have, because like the genome size of, and you have 200 such plates. Okay, so you have close to 60,000 colonies. Okay, now what you do is you take 100 colonies, put it in one tube. Okay,
the 100 colonies from the different plates at random.
At random. Okay, and you don't care who they are, you just do that. So this is your tube 1, another 100 colony, tube 2. This way, if you have 600 tubes, you will have all the 60,000 in there, but in bunches of 100 each, right? Now you keep a frozen stock from here, a little bit. You freeze it in dry ice and put it in -80°. Okay, the remainder you grow in a liquid culture. Okay, and when you are doing this kind of work, you are typically a postdoctoral scientist. You are supposed to have 10 hands and a few feet. Okay, so you will then see these are in bacteria, right?
Yeah.
So and it is an expression construct. So in every bacteria, one mammalian or yeast protein is being made. You don't know which one. So in this tube, 100 different proteins are being made. 100, let's just stick to yeast for the time being. Okay, so 100 different yeast proteins are being made here. 100 different yeast proteins are being made here. So in every of the 600 tubes, 100 different yeast proteins are being made. I am not at all claiming that the set that is being made here and the set that is being made here are mutually exclusive. I am not at all making that claim. Okay. Now you take all the cells that you have grown in a pellet. Okay, and let us not get into the detail. You deploy method to make a solution of all the proteins. Okay, so you will have such 600 solutions of proteins. Each will have 100 different, 100 proteins from yeast, right? Now let us assume that there is no other complication. Bacteria can make the functional protein. Let us assume that. So then in one of the tubes, or at least one of the tubes, of the 100 recombinant proteins, one will be this enzyme, right? So you will take 600 reactions. In each, you will add X plus, tube 1 extract, okay, or X plus tube 2 extract, right? And in each tube, you will also add radioactive phosphate. Okay, the phosphate will be radioactive. So you will know in which reaction X became radioactive, right? There are challenges there, but technically you can know, right, in which reaction X became radioactive. So whichever, if it is just 1, fine. If it is 3, fine. So those are the 100 within which the enzyme that phosphorylates X is present, right?
Yes.
But you kept a frozen stock in the very beginning, right? So now you go back to that frozen stock. No, I misspoke somewhere. Somewhere it is not following the logic. So when you pick the 100, you actually will pick from an arrayed library. not from a general library, from an arrayed library. We will come to arrayed library a little bit later. So you will know exactly which 100 you picked. Like every, say in a plate there are 96 wells, okay? And the wells can be numbered on, so I'll come to this slide. Oops.
Like, you can do undo.
Ram. Hare Ram. Did I? अच्छा ठीक है. So, sorry, everything remains the same, just this thing changes. So in a 96 well plate, you will have, like a 12 by 8 array, okay? And these are numbered A, B, C, D, E, F, G, H, and these are numbered 1, 2, 3, 4, up to 12, okay? So every well position will have a number. B3 will mean this one, right? E7 will mean A, B, C, D, E, this one, right? So you will know. So you make your collection of 100 in this manner. You maintain an Excel spreadsheet and you know which 100 went to each of the tubes, okay? So that is the only change. Everything else remains the same. So now you know one collection of 100 that...
Sir, sorry to interrupt. So like for one, the like single vial having 100 clones is distributed in this plate, or all the 600
No. So this is 96 plates. You will have 600 such plates.
Okay.
Yeah, 600 such plates.
So we are basically distributing small aliquots of this one vial into one.
Every well has a unique clone. Okay, so the way it is done is that this is your plate, right? After you have transformed with the cDNA library, this is how your plate looks like, right? So, and you have your 96 well plate here, okay? Then you take this one, put in here. You take this one, put in here. You take this one, put in here. Like that. But you have to do it for, like when I made this kind of library, it was done by a robot. Like for that one day, I was not considered a robot. Okay. So you make the library like that. But now you understand, right? So how you are collecting your 100. But you know which 100, right? Because you have an Excel spreadsheet that said that in the first tube this went. So now you take that and go to that 100 and then you divide it into 10 batches of 10 each. Okay? Now you do the thing all over again and see in which batch of 10 you get the phosphorylation. Then you just test each of the 10 in the third step and you get it. Like some fantastic experiments have been done like this using expression library and finding kinases like that. There are some very, very good papers that have been done like that. Okay, so I answered your question?
Yes, sir.
You more or less understood.
Yeah, but so like here we are, in the beginning only we are testing for an enzyme which phosphorylates, but somehow we do not know that what that enzyme does, like whether it phosphorylates or it like isomerase type function, or like if we do not know, then
if you know, so if you do not have an assay for the gene product that you are looking for, then there is no way. Either you will know the sequence and you are looking for a particular sequence, or you are looking for a particular function. Okay, you cannot randomly look for things.
Or
Okay.
like, through the whole process which we are doing till now, like after the plasmid isolation sequencing, we can use some bioinformatics tool to see whether we have some sequence similarity with any organism or not, and what is the function of, if we find a similar gene or similar sequence, what is the function of that. So from there we can detect, okay, so this may be the function of my new gene or new like
Yeah.
protein which I'm looking at.
So let me tell you my first PhD publication. So I used to, as I told you, that I used to work on protein synthesis. Okay. There is a translation initiation factor called EIF3. Okay. It is written like this: EIF3, eukaryotic initiation factor 3. It has 10 subunits. Okay. Now, today you have the genome all known, right? Every gene, every mRNA sequence, the whole genome sequence, everything is known. In 1995, when I started my PhD, and you were not born, then it was different. We did not know the genome sequence, right? So, how I did my job? So, we were very good with biochemistry. We were very good with purification of protein. I will not digress there today. Maybe one day after you come back to campus, I can have a casual chat with you. Forget about how I did it. I purified EIF3 to homogeneity, meaning I have a protein solution where I have EIF3 and only EIF3, nothing else. Okay. Then I ran a protein gel. Okay, you have a molecular weight marker here, and you ran a protein gel, and you get to see the 10 subunits of EIF3. Right? Then it is a protein gel, so the bands are resolved. So you cut each of these protein bands, and then you sequence the protein. So you get the amino acid sequence. You don't get the whole sequence, but you get some 15, 20 amino acids. Then you do reverse translation and employ the codon bias for the particular organism, because all the codons are not used in all the organisms equally frequently. And then you search a database. In those days, I will come to it next, we used to have what is called as EST database, expressed sequence tag database. What is EST database? It is nothing. It is just like your cDNA library, but the manufacturer, the company that would sell it, they will not guarantee that it has the full length, meaning up to AUG and beyond. So you have this mRNA, maybe it stopped over here. They are not guaranteeing that we have the whole thing. Okay, but because it is a cDNA library nonetheless, so it will always have this part of the sequence right after the poly-A tail. So these are called 3' biased libraries, because in the library, if nothing else, the sequence towards the 3' end of the mRNA will be there. Okay, so if your amino acid sequence that you got, if that corresponds to the sequence somewhere over here, you will find a match in the EST library. And then you figure out from where this EST came, if something else is known about it. Then the first gene that I cloned is called INT6. You can check under my name. INT means integration. Okay, so this is a gene that was originally identified in the context of genes that are important for protecting, in mouse of course, mammary gland from this virus called mouse mammary tumor virus induced cancer, breast cancer. So obviously I have no clue. I still don't have any clue that why such a gene is a bona fide partner of a protein complex involved in translation initiation. But I knew that Okay, so this is where I have to look. So this is, we have digressed quite a lot. I don't want to continue on it any longer, but this is how you would clone the genes in the early days. So once you know an EST sequence, then you make a DNA probe out of that and you clone a cDNA library. Okay, you screen a cDNA library, and that is what I need to talk about today, more importantly, because of this. So, who understood the concept of genome coverage in the class? Anybody would raise hand who understood the concept of genome coverage? हाँ आकांक्षा बोलो.
So does it mean like the slide which explains about the representation of the
Yeah.
Yeah, so what I understood from there basically is that not all mRNAs are produced like in equal quantity and always. So
Yes.
there is a chance that when we are making a cDNA library, so for example, there are some essential genes. So their copies, their mRNA copies will be higher than a gene which is expressed under certain conditions. So there is a chance that while making a library, we may miss those out. So to make sure that we have equal representation, like everything is represented well enough to make a library, we make sure that we like we have a number, we have like 10 to the power 5 to 10 to the power 6 clones made, containing the library. So that will ensure that we might also get the sparse mRNAs covered.
Yeah. So as all the mRNAs are not produced in equal quantity, so if I have, say we are talking about yeast has roughly 10,000 genes. So if I have in my collection of yeast cDNA library only 10,000 colonies, then chances are that of the 10,000, 5,000 will only be for actin, tubulin, and ribosomal RNA. So a rare gene being represented in that 10,000 will be very, very low. So the thumb rule that is used even today is typically to, because see, on the other hand, if you go up to 1 million, then of course every gene will be represented once, but you will not be able to handle it. It will be, unmanageable. So typically the compromise that we would make is, like 3 times the genome. Okay, so if it is 10,000 genes, 30,000 colonies I will deal with. If it is 23,000 genes, 1 lakh colonies I will deal with. It all depends on the subsequent application, but the minimum thing that you need to understand is that we should have more number of colonies in my collection for a cDNA library than the actual number of genes, so that the whole genome is covered at least once. Okay. Okay. So then, like if you think of my own experiment, so then you will have, see, in a typical plate, you will have 200 colonies, then only they are distinctly visible. Okay. So if you have 200 and you want to go for 10 to the power 5, so then you will have, almost 2,000 plates, right? Sorry, not 2,000, 500 plates. Okay. So when you have 500 plates, what you will do is that for each of the plate, you put a nitrocellulose filter on it and then lift it and then put the plate back in cold room. Okay. And you quickly fix the DNA on this nitrocellulose membrane by lysing it with alkali, and the DNA will get cross-linked to the nitrocellulose membrane. Then you probe it. In my case, I knew the EST sequence, right? So I will simply make a radioactive version of the same EST sequence, put it on the piece of paper, and if there is even one colony that made the DNA corresponding to my EST, it will be hybridized. and there I will see a black spot. So when I have the black spot, I will map this black spot with the plate that I have kept in the cold room, and I will figure out where the black spot came. The colony corresponding to that, I have not killed it, right? I have only lifted a little bit with nitrocellulose. Some colonies were, some bacteria were left behind. They would have grown by that time. I'll be able to locate that bacterial colony, pick up, sequence, and I will get my clone. Any question anybody has?
No, sir.
Okay.
Sir, regarding the linkers in the cDNA library, sir, do we have to add DNA ligase to add them?
See, it depends on at which stage you are doing it. If you are adding the linker at the RNA level, then you use RNA ligase. If you are adding the linker at the first-strand cDNA level, then you use a special DNA ligase that acts on single-stranded DNA. Because see, the problem is actually, just hold on. The problem is actually twofold. One is that providing a restriction site, but that's a secondary problem. A bigger problem is how do you make the second strand? First strand, very simple. You know that every mRNA has oligo-DT at its end, so you made a poly A tail for the reverse transcription, right?
Yes, sir.
There is no sequence consensus for the 5' end, right?
Yes.
So
Yes,
how
sir,
do
there
you
is no.
do the second strand?
So
Therefore,
we have to add some linkers.
yes, therefore the linker has to be added either at the RNA level or at the first strand cDNA level.
Yes, sir.
Someone else was asking something?
So I actually have a doubt when it comes to vectors. So what I wanted to know is like in the expression vector that you drew, there is a promoter sequence and all of that. So what I wanted to know is, can the cellular RNA polymerases, can they access the vector, the insert and all, like can they transcribe it?
So that's what is a shuttle vector for. You will have to provide the host specific promoter enhancer sequence.
Okay, but we don't have to provide the cell with the RNA polymerase, right?
No. See, you are making the protein in yeast, right?
Yes.
So if your polylinker, can you see my slide?
No, sir, I just see the blank screen.
Okay, just hold on. So now you can see it, right?
Yes, sir. Yes, sir.
So you will clone it here, right?
Yes.
And every example that you have seen, always it is a double digestion.
Yes.
So you know that which side will be the 5' end with respect to the mRNA.
Yes.
Let's imagine that it is on the left-hand side, like here.
Okay.
Okay. So in this sequence, if there is a yeast promoter, then your job is done.
Okay. Okay.
Right?
All right.
Okay. Any other question? कुछ नहीं, सबको सब कुछ समझ में आ गया? Very good.
Sir.
हाँ.
Sir, do we need to have the Kozak sequence in the vector?
Good question. The answer is no. Why no? Because Kozak sequence is immediately before AUG, right? Minus
yes.
1 or minus 3, I forget. So there is no way that, like, you know, your cDNA will not be expressed if the AUG is not there to begin with.
Yes.
If AUG is there, then the Kozak context will also be there.
Okay.
Right?
Yes, sir.
Okay. Like, are you convinced, or you don't sound convinced?
So I am thinking about if we if we go from mRNA to cDNA, in the cDNA, we doesn't ensure that in the cDNA sequence, in the 5' end of the cDNA sequence, the 5' end of the cDNA sequence will not contain the Kozak sequence.
Okay, so that's what I am trying to make the argument for. That is, let us take this one, huh?
Okay.
In this one, if this is where you have the AUG, okay, your reverse transcriptase either read the AUG or it didn't, right?
Yes.
If it didn't read the AUG, then whether you provide Kozak consensus in the vector or not, there is no translation of your genome.
Yes.
And if it read the AUG, it cannot happen that it only read up to A and just the nucleotide before was not read, right? So therefore, you do not, like providing Kozak consensus in the vector does not make any sense.
Okay.
But in bacteria, you have to provide the ribosome binding site in the vector.
Yes.
Anything else?
Hello, sir. Is there any method to avoid the secondary structure of RNA, mRNAs?
Yes, there are methods. The most popular one, reverse transcriptase generally works at 42 degrees centigrade. Okay.
Yes,
there is one reverse transcriptase that works at 50. Okay.
Thermo Fisher.
Huh?
Thermo Fisher one. Sir, I have searched for the efficient reverse transcriptase.
So,
which works at 50 to 55 degrees centigrade.
So that is one easy trick that people use, but it is expensive. So you have to have the money for it. And there are some old nanny recipe. Some people use mercury chloride, etc. Like one day I will tell you my sad story, what I had to do. So all of those things can be used to melt the secondary structure to some extent, but even after all of that, there is no guarantee.
Okay, sir.
Sir, and for that problem, like if the restriction site is also in the reading frame, then what we can do?
Honestly speaking, you would not even know that you have missed something.
Yes, yes.
That is the biggest problem. But you know, like the same problem is attacked by many people from multiple different angles. So eventually it comes out, but people have gotten frustrated that way.
Yeah.
Okay. Now, okay, I am sharing my screen another time. Now I will show you something that you need to follow very, very carefully. Okay. Can you see I'm trying to go to a website?
Yes, sir.
Just because I am trying to show you, it is not loading properly. Called Murphy's rule. Oof. I am searching the sequence for my new favorite gene. Okay. Okay, this is an mRNA sequence, okay?
Yes, sir.
The way to read it, this, this is 5' UTR in small letter, okay? You check, you will not find any ATG there. You should go to UCSC genome browser, study some 20 genes. So in the 5' UTR, you do not see any ATG, right?
So there is one ATG.
Either
one ATG is there.
Second line
After multiple Gs.
There is one ATG there, right? So this is called upstream ORF. Sometimes they will be there, but very soon there will be a stop codon. So you have to now figure out where the stop codon is. All stop codons start with U?
Yes, sir.
So there will be, is this a stop codon, UAG?
Yes,
is this
sir.
a stop codon?
Yes, sir. Yes, sir.
Yes, sir.
So sometimes there are these small upstream, let us see if it is in frame, okay? So if it will be in frame, there will be exactly multiple of 3 in between, right? So 22 letters there. Are Ramda, it is not, no, I have to, वो किधर से मिलता है?
So color change.
No,
Word
no.
count.
Word count. So it is 8 here and it is 31. So there are 39 nucleotides between the ATG and the TAG, right? So it's a multiple of 30, so it will stop there. So what happens is that sometimes you have this short upstream open reading frame, which down-regulates the level of translation. Okay, you can read up on Google that what short upstream open reading frames do. Some proteins have that. Okay, so that's why I'm saying that just play with a few genes and see, how they are. Now this is your ORF. Okay, up to here. Okay, now the job I have given you is give me sequence of 2 primers with which I would be able to amplify the ORF and clone it. Who will do it? You have to design 2 primers to amplify the ORF and clone it. ये HP कौन है? Who is this HP?
Me, Oishi.
अच्छा, चुपचाप पुस्तक. Ruchira, would you want to try?
Hello.
Yeah.
I don't. So we need a primer for the ORF. So we need to start with like the starting sequence, like first.
No goal. You have the whole thing. You tell me. You want me to go to this end? I have gone to this end. You tell me the primer sequence. Wherever you will ask me to go, I will go there. You have to give me the primer sequence. Okay, I will make your life a little bit easier or difficult, whatever you call it. Oh. This is rest of the sequence. I am not putting it there.
Okay.
Oops. Here, it's your whole mRNA sequence. Now you have to design 2 primers to amplify this.
So, but if it's mRNA, first we need to like convert it into DNA. If it's an mRNA
This is
sequence.
actually the cDNA form that is given. Na? You are seeing it.
Yes, yes.
Wasting time. Bolo. No. I want Ruchira to try.
So from the first part, if we need to create a primer like without like adding restriction enzyme sites, so then we can directly add like a opposite sequence to like ATG, CAC, the one which was provided.
Bolo. You just give me the primer sequence. I will write it. As you say, I will type it.
Okay, so one of them can be, so it will be TACG.
TACG. अच्छा TACG. TCGCC. TCCC, right?
Yes.
Never write DNA RNA without 5 prime 3 prime. Okay.
Yes.
Now you tell me. Do not give me goal answer. Should I write 5 prime near the T or near the C?
So the 5 prime should be near the T only, but then then we'd have to this would not be the primer, then the primer would be from the other end. I forgot to ask which was 5 prime 3 prime.
अरे जो पूछने का क्या है, like wherever is ATG has to be 5 prime in terms of cDNA.
Yes, sorry, sir. So it would be like T, C, A, from that side it will be the
Huh?
primer. Like the primer would be from other side. Like we need to start have the primer from 5' end, so we need a primer of the like the ending region, not from this region.
You just dictate the primer sequence to me. I will type it here.
Okay, so the primer sequence needs to be T, C,
T, C,
A,
A,
G,
G,
T,
T. Okay.
mm, and
C, C,
CC, and again T,
T,
and
C.
CG,
ठीक है.
and yeah. So this
Okay.
is like in
Now,
5' too.
where should I put the 5 prime? Where should I put the 3 prime?
So T should have the 5'.
This is your 3 prime, right?
Yes.
So in reality, you are saying it will be like T, C, A,
Yeah.
like this. This is
Yeah.
5 prime, right?
Yes.
And this is 3 prime.
Yes.
You are absolutely correct, Ruchira. So that is one primer. With one primer, you cannot do any amplification, right?
Yes.
What will be the second primer?
So the second primer should be ATGCA only.
Absolutely. The second primer should be the, so in our parlance, we call it the forward primer.
And
And
the
this
reverse
is called the reverse primer. The forward primer will have the sense sequence. Reverse primer will have the complementary sequence.
Yes.
Okay?
Yes. Yes.
But do you realize something as simple as requires thinking?
Yes.
Okay, it cannot be done mechanized manner.
Yes.
So if I will give you this, I will give you this kind of a sequence, and then I will ask you to design primers to clone it in a vector. Vector ka naam bhi de dunga.
Okay.
You will have 2 hours to research. Okay? Therefore, I want you to play with this. Okay? And your first quiz will be, Viva Bosi will be next Saturday and Sunday.
Okay.
Which is not next, meaning not really next, 3rd and 4th October.
Okay.
Okay?
Okay, sir.
But घबराने की कुछ नहीं है, ऐसा ही सब चीजें देंगे, हाँ? Absolutely nothing to be worried about. There will be maybe 4 or 5 memory based questions, but majority will be this kind of questions. Okay, like designing a screen, designing a the cDNA library, etc. So now the problem is that I needed another discussion hour to complete this. And what might happen is that you may have to find a new instructor for your course, because there is a very good chance I will be dead tomorrow. Okay, so if I die on 26th, so you will have to find a new tutor, instructor. So what will actually happen is that tomorrow I will have to start at 1:30, meaning 24 तारीख रात के 12 बजेगा, उसके बाद 1:30 बजेगा. I will have to start for JE, and then I will have to, the flight is at 6 a.m. It will go to Mumbai, and from there the next flight is at 2 p.m., which is typically late by 2 to 3 hours. So I might reach my hotel around 7-8 p.m. So जिंदा रहने का chances बहुत कम है. But if by some reason I am alive, certainly I cannot take your discussion hour on Friday. So if I am up to it, will you check, and will it be possible for you to sit down for the discussion hour on Saturday?
Saturday night.
Huh?
Saturday night, if after 8, it is okay.
After 8 only.
Okay, sir.
because you need
Yes, sir.
another discussion hour for the topic that we need to cover, right?
Okay, sir.
Otherwise, you will be in trouble. Huh?
Okay, sir. Yes, sir.
Yes, sir.
Saturday, 8 p.m. we can sit if
Okay.
nobody
So
else
Okay,
has
please,
sir.
any class.
please block your calendar. And more importantly, block your calendar. Uska time abhi. Oishi, please take a note of it. Uh,
Yes, sir.
I need to fix the date for the Viva, date and time for Viva C. So it will be Saturday and Sunday. So let us say from 10 a.m. to p.m. for half the batch on 3rd September, 3rd October. Is that okay?
So from 10
Yes, sir.
a.m. to what?
2 p.m.
Okay, okay.
Your engagement is only for, like at max 15 minutes. It is me, I will have to sit through everything, the whole thing, but individual student, at max, you will be spending 25 minutes. Like you will come, be in the waiting room, where the previous student finishes, you come in, your interview time from BioOC time will be 15 minutes, and you leave, okay? So
Okay,
rest
sir.
of the day, you can watch your movies and everything. I have no problem with that. Is it fine? कोई कुछ जवाब नहीं दे रहे हैं.
Yes,
Yes,
sir.
yes,
Yes,
sir.
Yes,
sir.
sir.
Okay. Similarly on Sunday, okay, depending on how much we have progressed, Oishi will prepare a timeline. So Oishi is my PhD student. She is the tutor for this course. Oishi, you should formally introduce yourself to the group by sending an email. She has terrible internet connection. That's why she is not more prominent. So she will share an Excel spreadsheet with each of your, interview time, and I will do it in that sequence, and I will allow you in and out, and I will share the question papers uh, only to those, 6 students. The first question paper will be shared. The next 6 students, a different question paper will be shared, like that, so that there is some semblance of fairness. Okay, like no matter what I do, it cannot be absolutely fair, but some semblance of fairness.
Sir, so those students who cannot be present on Saturday will be accommodated on Sunday for the Vivos.
Like I will prefer to have it done in a particular sequence. If you are not available on Saturday, please immediately put it on WhatsApp group so that she can take care of it.
Okay, sir. Sure.
Okay. So
Okay.
आज के लिए बंद करें?
Yes, sir. We can.
Okay. तो ठीक है. Have a good night, all of you. I am hungry, famished. I need to go for my dinner. And I will share the recording. But you understood what you have to do, right?
Yes, sir.